goes into the gallbladder and the liver, and then eventually gets shipped up to the brain along the vagus nerve. So I think that's what's going on. And so it accumulates in the brainstem nuclei, and then you get nasty conditions like Parkinson's. Probably even contributes to autism.
Hello and welcome. I'm Sarah, and in this episode, I have the pleasure of talking to senior research scientist, Dr. Stephanie Seneff. Now, Dr. Seneff is a polymath, and she's very humble, so she's not going to like it that I said that, but she's published over 200 peer-reviewed journals on a wide variety of subjects, and she's also written 2 very well-selling books. One of them is on autism and a variety of different causes of autism, and then the second one on glyphosate. So the book on glyphosate goes way beyond just a weed killer. It educates the layperson on lots of other health matters. Same as this podcast, it's for lay people as well as people who are way ahead. So Dr. Seneff hasn't dumbed things down today, but there are times when I've asked her to explain something that's complicated so we can all keep up. So in the podcast, of course, we're going to talk about glyphosate a bit, but it also ties into how glyphosate actually affects people. And this links into things like serotonin, tryptophan, and melatonin, but also how it affects the gut and glycine. And we also talk about other hormones such as melatonin and vitamin D and their complex roles in the body, and whether supplementing is a terrible idea or really important. As this is a quantum biology channel, we are going to be diving into deuterium, things like proton tunneling, but Dr. Seneff's going to explain anything that I think people might get lost at. I've also made a video on deuterium on this channel, so anybody who's never heard of it before, I suggest watching that. We're also going to discuss methylation, because this is important for a wide variety of aspects of health, from detox to making DNA to making neurotransmitters and a lot more. And Dr. Seneff's got a very interesting take on methyl groups, and it ties in with the deuterium and the glyphosate story. Throughout the podcast, we talk a lot about sulfate. So this is a very important mineral, but towards the end of the podcast, I wanted to talk about other minerals such as iron, iodine, fluorine, and manganese, the ones that don't get talked about so much. So Dr. Seneff explains how excesses and deficiencies can arise and how these can be harmful for health. So there's a lot in this podcast, and I just want to thank her for being so generous with her information. All of the links to books and such like are in the description. Thank you very much for coming on the channel, Dr. Seneff. Would you like to introduce yourself so that people understand why we're going to have such a diverse set of topics today?
Okay, sure. Yeah, I'm Stephanie Seneff. I'm a senior research scientist at MIT, and I've been at MIT all my adult life, starting with undergraduate training. I have 4 degrees from MIT, a bachelor's in biology, master's EE, and PhD in computer science and electrical engineering. And most of my career was spent developing software to support human-computer dialogue, precursors to things like Siri and Amazon Echo. But about 2008 timeframe, I got very interested in autism because I was seeing the rates go up, and I wanted to figure out what was going on. I mean, it was considered to be a genetic disease, and most of the research dollars were going into figuring out the genetics of autism. I wanted to figure out the exposure problem and how that links to autism. And so I started doing that in 2007, looking at various toxic exposures that were looking for things that were also going up in step with the rise in autism. And took me 5 years before I discovered glyphosate. And glyphosate is the active ingredient in Roundup, which is a pervasive herbicide, the most used herbicide on the planet, kills all plants except those that are engineered to resist it, and is completely harmless to humans in theory, but that's not true. And so this is my book, Toxic Legacy, which was published in 2021, how the weed killer glyphosate is destroying our health and the environment. And that book goes into great detail about what I understand as the biology of how glyphosate works, its diabolical effects on all life.
I've read the book, and weirdly, my favorite chapter in it was the one on sulfate. And I think we've got so much to talk about, and I want to bring up things which we neglect. And I think sulfate will tie in really well with the glyphosate, well with aging, and really well in with cardiovascular disease, hormones, and neurotransmitters. And obviously, you're the expert, so do you want to take over here to introduce the sulfate story to people?
Yes, sulfate really caught my attention early on. When I first started looking at autism, I came across some work by a woman named Rosemary Waring, and she had published in the 1990s. She was an expert on autism. She treated several autistic kids, like 80, I forget how many, but a lot of autistic kids were under her treatment. This is at a time when autism wasn't that common. And she was doing a lot of measuring of metabolites in the urine. And she discovered that these autistic kids had extremely high levels of sulfur metabolites in their urine, much, much higher than the controls, like 100 times as much type of thing, I mean, completely out of line. I think it was thiosulfate and sulfite, those two sulfur-containing compounds. And sulfate was depleted in their blood. They had low sulfate in their blood, and they had these other sulfur metabolites in the urine. And she figured out that must mean they've got a problem with sulfate handling in the body, some kind of defect in sulfate. And she even proposed that they could have a defect in an enzyme, class of enzymes that add sulfate to biologically active molecules, and in particular polyphenols. So she had a very specific idea. She didn't prove it, but she suspected it, that these kids could have a defect in an enzyme in the gut that attaches sulfate to things that are aromatics. So the aromatics have these rings, and you attach sulfate to them. And then when you attach sulfate to things with rings, biologically active molecules with rings, carbon rings, usually, other things can be in there, but mostly carbon. You attach the sulfate, it makes it water soluble. So many of these things are fat soluble. Adding sulfate makes it water soluble, makes it able to be transported freely in the blood, and is very important for delivering it to wherever it needs to go. Both things that are useful and things that are toxic. You can the liver attaches sulfate to things that are toxic, making them water soluble, making them able to travel through the blood to the kidneys and get sent out. And so it's both to excrete toxic molecules that are not water soluble and to transport them. And so it's the aromatics, which are, I can get into what they are, but that's a huge class of biologically important molecules. And then also the sterols. There's a whole other set of sulfation. These are called sulfotransferases. And there's a whole bunch of different sulfotransferases in the body. And they mostly attach sulfate to these two classes of molecules. The sterols, which is cholesterol, vitamin D, the sex hormones, that's the sterols. And then you have on the other hand, the aromatics, which is the neurotransmitters, and also the aromatic amino acids, and also various metabolites that are produced by the microbes in your gut. So all of those things routinely get sulfate attached to them for the purpose of being able to, I think, to deliver sulfate throughout the body. That's something that people don't realize, but I believe that's true. They're being used to deliver sulfate, and they're being used, of course, to get around, solubilize them, making them able to move in the blood. So those are really important points around sulfate. And when you have a deficiency in sulfation, you've got a huge problem biologically, systemically. And it's interesting because just a couple of years ago, a new paper was published looking specifically at autism, looking specifically at sulfotransferases and hitting gold. I mean, finding out that these autistic kids had tremendous deficiencies in sulfotransferases in the gut, in the blood, and in the brain. Quite amazingly, they actually had some post-mortem data on some of these kids looking at the pineal gland, and they found severe deficiency in heparin sulfate, which is a sulfated molecule that lines the blood vessels. They found severe deficiency in heparin sulfate in the brain, along with deficiency in the sulfotransferase that makes the heparin sulfate. So it all fits together really well. And so I think sulfation deficiency is a core factor of autism.
That's fascinating, because I think for people also, they don't realize that sulfur and oxygen are related, because I didn't know that the mitochondria used hydrogen sulfide, but that's massively interesting. But also autism is very high on the sort of scale of a severe disease. What about things in between? Because there must be other things where sulfate deficiency is a problem, because I asked you before we started recording about other mental health issues like bipolar depression, and then we were talking about neurodegeneration, but then lack of sulfur must be key for all sorts of things. What else would there be?
Yes, well, for example, serotonin, I could get into glyphosate more specifically here, because serotonin is a neurotransmitter. It's made mostly in the gut. Most of it's made in the gut. It comes from tryptophan. Tryptophan is an aromatic amino acid. So there are 3 of these aromatic amino acids. And I go into great detail about this in my book. tryptophan, tyrosine, and phenylalanine. So tryptophan is a precursor to serotonin. The gut microbes make the tryptophan using a pathway called the shikimate pathway, the shikimate biological pathway. That's the pathway that glyphosate famously disrupts in the plants. That's how it kills the plants. They've determined that it disrupts the shikimate pathway. That means the gut microbes can't make tryptophan when they're just exposed to glyphosate. And also, of course, the plants can't either. So tryptophan becomes deficient in the diet and in the gut. Gut microbes are making all kinds of things for the host, and tryptophan is one of the things they make. Our cells can't make tryptophan. It's called essential for that reason. And they can't make it because they don't have that shikimate pathway. So Monsanto was the maker of glyphosate, and they said, oh, don't worry, it disrupts this pathway in plants, and humans don't have that pathway in any of their cells, so it's harmless to us. That's the argument. But when you think about how the gut microbes are using that same shikimate pathway to make these critical nutrients that we can't make, we start to realize how crucial that could be and how it could disrupt our health. And so serotonin becomes deficient because of glyphosate exposure in the gut. And then serotonin also needs to be sulfated in order to be transported. So the gut sticks a sulfate molecule onto the serotonin. Sulfate, by the way, is sulfur, 4 oxygens and -2 charge. Sulfate, SO4 -2. And it's stuck onto these, every one of these serotonin molecules gets a sulfate stuck onto it before it gets shipped out. And it depends on that sulfate to deliver it to the brain. So not only is it deficient, it's also unable to be shipped in adequate amounts to the brain. And so the brain becomes severely deficient in serotonin. Serotonin is a precursor to melatonin. So that means melatonin is also deficient. Melatonin deficiency leads to sleep problems, and we have an epidemic. Sleep is going up. Sleep disorder is going up exactly in step with the rise in glyphosate usage over time. I've looked at that, and I've got the data. Perfect match. Very, very many different conditions that are going up dramatically match perfectly with the rise in glyphosate usage on corn and soy crops in the United States. I've done a whole bunch of analysis of that, and together with Dr. Nancy Swanson, she and I collaborated on a number of papers where we've shown this data, and sleep disorder is one of them. It makes sense because melatonin is deficient, and also melatonin gets sulfated in the pineal gland before it gets shipped into the cerebrospinal fluid. So there's another problem. When you've got sulfation problems in the brain, not only can you not make enough melatonin, but you can't sulfate it, and therefore you have a tremendous deficiency in the delivery of melatonin sulfate to the brain while you sleep, which is absolutely crucial for the sleep to be able to do what it does, which is to help clear the garbage.
Oh, yeah, definitely, because melatonin does a lot more than just sleep. It sort of detox and apoptosis, and it regulates other hormones as well.
Yeah, melatonin is a very interesting molecule all by itself, and I've read some fascinating papers about it. In fact, individual cells all over your body I didn't only learn this recently. It's an interesting little nerdy fact, but they make melatonin, the cells, and then they break it down. So they're actually using it themselves. And we can get into this later because it's related to the deuterium problem that we'll get into later. But the melatonin is actually made by all the cells and utilized by themselves. They don't share it with anybody else. They don't put it into the blood. They just make it and break it down.
That's really
and they're doing that in order to supply deuterium depleted water to the mitochondria. So we can get into that later. That
I love deuterium because the mitochondria make melatonin as well, don't they? So
yes,
and it's really going to tie in later. Because one question, somebody might say, well, surely you can just take tryptophan orally to make up the deficit. And then also, secondly, you've touched on, because I'm not a fan of melatonin supplements unless somebody's got cancer. So would you say that they don't work properly because they're not sulfated? I know I've asked 2 questions at the same time, but I'll forget.
It's actually more complicated than that. Yeah. I'm really fascinated by melatonin, and because I love biochemistry, and I've learned some of these reactions. I don't know them perfectly, but I do know that the process of making melatonin, originally from tryptophan, involves adding 2 things to it: a methyl group and an acetyl group, acetate and methane. They get stuck on to the original tryptophan molecule to make the melatonin. Both of those, the acetyl and the methyl, are very valuable to the body because they are deuterium depleted. We have to get into deuterium. I'll explain. Maybe I should take a a bypass into deuterium to give people a sense of what that is.
Yeah, maybe.
I should do
I
that
think
now.
it's one of those things we can come back to. Can you take extra tryptophan to overcome glyphosate, and then this? Because some people know what deuterium is and other people don't. And I think it's really important, because especially when we talk about aging later, because it's missing out of the pillars of aging. But I'll let you
yeah, well, so let me tell you a certain bottom line, which is I'm worried about melatonin supplementation in terms of how the melatonin came to be. And I know that they've developed a lot of chemistry, not biochemistry, but physical chemistry in the laboratory to make various biologically active molecules. And so I haven't been able to figure out, but I have a feeling the melatonin that they make, that you take when you take a melatonin supplement, is not natural, that it's synthetic melatonin made in the chemistry lab. I haven't confirmed that. But if that's true, the acetyl group and the methyl group will not be good because they won't be deuterium depleted. So there's a whole other aspect of taking supplements that you get into when you realize this importance of deuterium. I can give you another example, which is the PUFA, polyunsaturated fatty acids. And lots of people say, oh, yeah, they're really good for you. We need more. Take them, I say, no, don't take them, because they are actually biologically, they're susceptible to oxidative damage. And when they get oxidized, they do all kinds of bad stuff. Your body needs them in the cell. But what it really wants is the precursor, which is a completely saturated fatty acid. So the saturated fats are the best fats to eat. This is like so counterintuitive. They have low deuterium hydrogen. Their hydrogens are low in deuterium because of how they're made. And then they can be turned into the polyunsaturated fatty acids in the cell in order to supply the mitochondria with deuterium depleted water. So it's really, really interesting and intricate. But you start to realize, I don't like taking melatonin. And in fact, if someone really wants to take melatonin, I would say take tryptophan, because the tryptophan will get converted to melatonin in your body. And those methyl and acetyl that I mentioned will be good guys. They'll be made by the gut microbes in such a way that they'll be deuterium depleted. Whereas if you get it from a synthetic lab, they don't pay any attention to that.
And also, getting melatonin from your gut isn't naturally where it comes from. It comes from your pineal gland, and also there's the photoactivation of all of these molecules. So there's all sorts wrong with supplements. But I think we'll kind of move on, because people who don't know what deuterium is,
we better get involved with deuterium at this point, because they're probably completely mystified. Yes, it's really deuterium really grabbed me, and I have to say it's only been since December of 2019. It's been quite a journey for me, because I've discovered all these things, and they open up a huge window of a whole new space, and then I have to work that into the grand theory. So it's just been, I love a puzzle, and this is a gigantic puzzle. But once I learned about deuterium in December of 2019, it was Professor Laszlo Boros, who sent me a blind email saying, oh, I read your article. I wrote an article with Greg Nye, and was on metabolism, and it was talking about sulfate and its disruption and what that causes in terms of all the stuff that's going on in the body. He liked that article. He said, he basically said, I loved your article, by the way, deuterium, So I'm like, oh, what's that about? So he really kind of tickled my interest. And he and I have now been we've been in correspondence, and even I've given talks at his, that he's hosted and things like that. So we're now engaged with each other, and it's been really fun. And he's just taught me so much, and his just opening up that whole space. And of course, then digging through the research literature to try to understand more. And it just was a gold mine for me. I immediately was attracted to it because, for the simple reason that I already knew which enzymes I suspected glyphosate was messing up. And I have specific reasons, and you know that from my book, why glyphosate would target certain enzymes. And then it turned out that a whole set of enzymes that are targeted by glyphosate are crucial for maintaining low deuterium in the mitochondria. And that's the key. The body needs to maintain low deuterium in the mitochondria. What is deuterium? It's heavy hydrogen. So hydrogen is the smallest atom, one proton and one electron. Deuterium has one proton, one electron, and one neutron. So it's heavy hydrogen. It's twice as heavy as hydrogen. Hydrogen is by far the most common atom in the body in all living forms. And deuterium is an isoform of hydrogen, just like carbon-14 versus carbon-12. It's an isoform of hydrogen, and it's not radioactive, but it is very, very different in its behavior from hydrogen. And of course, hydrogen is absolutely crucial in all of biology. All these reactions that take place almost always involve hydrogen, in some way. Often it's hydrogen and oxygen working together to do stuff. And since deuterium behaves very differently, when you hit a deuterium at a place where it's supposed to be hydrogen, the enzyme can get stumped. It cannot work well. And in particular, there's a particular enzyme, which is ATPase. And that's the enzyme in the mitochondria that converts, that makes ATP. When you metabolize sugar, you make ATP. ATP is the energy currency of the cell. And the enzyme that does that doesn't like deuterium. That's really a big red light, doesn't like deuterium. And so all these enzymes and all these proteins and enzymes have come around to figure out how to channel hydrogen, but not deuterium, into the intramembrane space of the mitochondria. So it's super fascinating in terms of the biophysics and the biochemistry. And there are these enzymes called flavoproteins in particular. It's a whole class of enzymes, flavoproteins. And they have a very interesting skill that is to say they can funnel hydrogen through a channel that they create such that deuterium doesn't fit. So it's basically got very skilled design of these proteins to facilitate the movement of hydrogen from one place to another, and also to say, no, deuterium, I'm not taking it. So if the molecule that's coming in has a deuterium where it should have a hydrogen, it won't react. It'll just throw it away and not do that one. So it's able to select the instances of that molecule that have hydrogen in the place that it's going to grab it from. They make sure that it's hydrogen. And so they can deliver hydrogen to the product.
Is that why the Krebs cycle has got so many steps? Because it's just checking.
Absolutely, absolutely. Step, and also glycolysis. Have you looked at glycolysis? It's so interesting. It seems like it shouldn't be that complicated. Glycolysis is what's happening anaerobically in the outside of the mitochondria, in the cytoplasm. When glucose comes in, I'll make this as simple as I can. Glucose comes into the cell, it gets converted into pyruvate. Outside of the mitochondria, it gets converted into pyruvate. And then the pyruvate gets fed into the mitochondria to make the ATP. And so the part that converts glucose to pyruvate has all kinds of fancy steps. It's got a lot of steps, and it doesn't look like it needs all those steps. But there are steps where it has these enzymes called isomerases that they go back and forth between two different molecules that have the exact same, all the atoms are the same, but they're arranged a little differently in the molecule. So you're sort of taking an H off here and putting it over there. And what's really fascinating is that these isomerases are able to shed deuterium. So what's happening is they're going back and forth between this one, these two. And if there's a hydrogen, if there's a deuterium, it's going to get thrown into the water, and you'll pick a hydrogen instead. So by the time you're done with this shifting back and forth, you end up with hydrogen. So it's a way to clear the deuterium, put it into the water, which is the water in the cytoplasm, not in the mitochondria. So the cytoplasm doesn't mind having extra deuterium in its water. But now you've got this molecule.
Bind to hormones and cholesterol and stuff like that and inactivate them.
Say it again.
Doesn't deuterium bind to cholesterol and hormones and things as well and disturb them, or is it only destructive to the ATPase in the mitochondria?
Yeah. Well, anywhere the deuterium goes, for example, methyl groups is a very interesting one. And I can get a whole detour here on methyl groups. This is like, it takes you a lifetime to figure all this out, but it's so fun because this gets back to the gut microbes. I told you methyl and acetyl, right? Those are the two things that are stuck onto the melatonin. I think all of those, serotonin, melatonin, dopamine, all of those, the sort of neurotransmitters that come out and hormones that come out of the aromatics. So the aromatic amino acids are those three: tryptophan, tyrosine, phenylalanine. They come out of the shikimate pathway, which glyphosate disrupts. So we've got problems right there. Then they get sulfated in order to get pushed around, so they've got a problem there. And then furthermore, those things get metabolized with enzymes that know how to pick hydrogen over deuterium. And they also get things attached to them that they know have low deuterium. So the acetyl and the methyl, they're really, really important. And there's all these acetylation pathways, methylation pathways in the body.
Yeah, like the histone acetylation is what people might know about. So we're now getting down to gene regulation being messed up by the sulfate and the deuterium. But yes, I'll let you carry on.
Right. Yeah, it all gets really complicated. I have it all in my head, and it's kind of hard to know what to tell first and what to tell next because it's so complicated. I'm hoping to just interest the audience into learning more because fascinating space. It can really occupy a lot of time if you get involved with it. But so I was aware for a long time. I was like, why is there such obsession on methyls and acetyls? You know, like they're moved all around, all over the body, put here, take off, put it over there. You know, the methylation pathways, they methylate the DNA. You methylate the proteins. You know, you can add these things to various big biologic molecules that are involved in life. There's ways to sort of stick them there and then take them off and then stick them somewhere else, sort of move them around. You know, so the body seems to obsess on methyls. And why is that? That was something that I was intrigued by long before I knew about deuterium. Why are methyls so important? This sort of one carbon metabolism, they call it, you know, where these methyls, and the methyls have really powerful roles in changing behaviors of cells. They can really change it. That's the whole epigenetics, you know, methylation of the DNA, which is also tied to autism, is disturbed methylation of the DNA associated with autism. So the methyls are really important. Well, why is that? And I think I have a unique answer, which is that the methyls have really low deuterium. And then the reason why that's true is really, really interesting because it goes back to the gut microbes. And the gut microbes, one of the things they can do is they can take hydrogen off of molecules. You know, they have hydrogen stuck to carbons all over the place in these molecules. They can take two hydrogens off and put them together and make a hydrogen gas, H2.
Oh,
It's a gas.
okay. Like, yeah, molecular
Right?
hydrogen. Yeah, because people take molecular hydrogen tablets, but I knew the gut microbiome could make molecular hydrogen as well. But
That's right. And that is super, super important because I found an article from the 1960s, I think. This is something that was studied, you know, in the past, but we've sort of lost track of it. 1960s, there was an article looking at a microbe and looking at its ability to make the hydrogen gas and discovering that the gas it made had extremely low deuterium. It was so the normal amount of deuterium in water, for example, it's a natural element. It's all over the place. It's in seawater at 155 parts per million compared to hydrogen. And but in this hydrogen gas that these microbes made, it was only 20 parts per million, way down compared to 155.
I bought some deuterium depleted water that was 25 ppms because just so people can like get a scale, we're meant to be about 120, aren't we, to be healthy, and anything over 180 is bad, and seed oils are things like 200, so 25 parts per million is very low.
Yes, and there are foods that are low in deuterium, and one of them is saturated fat. Saturated fat is probably the lowest of all the foods that I've looked at. Lowest amount of deuterium is in natural saturated fat produced by animals that are fed organic food, you know, because if they're not fed organic, then the deuterium goes up because they're being disturbed by the glyphosate. So organic saturated fats are the healthiest fats. And I think a big reason for that is that they're low in deuterium.
What about coconut oil? Because that's saturated but not animal, and then cacao butter is saturated but not animal. Are they high or low in deuterium?
I know that coconut oil is, and I suspect cacao butter is as well, because I think any kind of saturated fat, I should say both animal and vegetable, I suspect. But what I've seen, there's not very much information on how much deuterium is in various foods. So I'm relying on some stuff I think that Laszlo showed me, and I think even Laszlo may have done the work, but they haven't done enough looking to see what's low, what's high. But in general, fruits are high in deuterium, and fats are low, and saturated fats are the lowest. And alcohol, interestingly enough, ethanol produced biologically is low in deuterium. Which is also interesting. So yeah, could be a reason why vodka is so popular.
Yeah, I quite like a vodka and a rum now and again. So that's, you know, yeah, alcohol is a funny two-edged sword kind of creature. Yeah, I didn't know that. What because people sometimes get worried about deuterium, and I know that you can get the deuterium depleted water, and then you've mentioned different foods, but then I don't know anything about the glyphosate and the deuterium link.
Yes, so let's keep going then, because we haven't quite got to the methyl yet.
Yes.
We say the methyl group and we say the hydrogen gas. What's the connection? The gut microbes are making the hydrogen gas. It's super low in deuterium, so it's extremely valuable. It's in your gut. There are other microbes that take that hydrogen gas and turn it back into organic matter. So there's a whole process in the gut that goes on, making gas and then turning the gas back into organic matter. And so the hydrogen gets converted into methane by these methane producing bacteria. Now methane is CH4, so that's got 4 H's. It came from hydrogen gas. tremendous molecules, CH4, all hydrogen, almost guaranteed, right? 20 parts per million instead of 155. Much, much less likely to be deuterium. And that's the methane. So now what happens to the methane? Well, we talk about, the cows make methane gas and
I
it
was just going to
causes
say
global climate change.
that
Right?
farts are really low in deuterium then. Cow farts.
Farts are low in deuterium,
Yes,
right?
yeah.
And breathe them in.
Yeah. Yeah.
And,
Yeah.
but then the methane actually then normally gets converted into methanol and then formaldehyde and formate, and then ultimately gets burned and produces carbon dioxide and water. So that whole thing goes all the way around. So you have the hydrogen and you have the carbon dioxide, and you put them together to make the methane, releasing some oxygen. That's this whole, these gut microbes can do that. They've got the methane, CH4. And then when it gets converted to methanol, it's no longer a gas. And then the formaldehyde and formate, that's no longer a gas. So those are going to be nutrients that are produced from that hydrogen gas. It has to go through that gas cycle in order to be gold. That's what makes these the methyl groups are methane minus 1C. It's just CH3 instead of CH4. And they're attached to various organic molecules. That's this whole methylation pathway stuff. But those methyls, the body keeps track of them. It's like, oh, this is gold. This is a methyl that came from the hydrogen gas. It's gold. Hang on to it. Make sure you keep it and keep it and keep it, and then finally metabolize it. So the methyl that's attached to the methionine, for example, has this golden methyl, and methionine is a sort of universal methyl donor in a way. The methionine and the tetrahydrofolate, all these different molecules that are very involved in the methylation pathways, they got those methyls from a good place, originally from the hydrogen gas. And so the body kind of knows that they're important to keep. And then you eventually, and so I actually had a hard time figuring out what happens to the methyls once they get on the DNA. You have DNA methylation, which is all part of epigenetics, and how different methyls in different places have different effects. It's very complicated with all these different signaling processes that go on with the methylation. But it took me a long time to find out how the methyls get taken off, because you think, well, there's the guy that puts the methyl on, the methyl transferase. There's got to be a guy that takes the methyl off, right? And I couldn't find it. I was like really puzzled. And I finally figured out it's not taken off. It's actually metabolized in place. So that methyl eventually becomes formate and then eventually gets taken off as carbon dioxide. It's actually getting metabolized in the mitochondria. And because it's gold, it wants to get to those hydrogens that are on that methyl group. And so it's quite fascinating, the biology of methyl groups. The body hangs on to them as sort of reserve energy. And then when it has a chance to metabolize it, it's going to deliver hydrogen to the mitochondria. That's low in deuterium. You see what I'm saying?
Oh no, definitely, because it's like there's more that it's more than one way to control the deuterium and methyl and hydrogen problem. I was just going to ask, but when we detox molecules, don't methyl groups get added to certain molecules as they get removed from the body, or is it only very, very nasty molecules that get methylated because it's a priority to get them out?
Well, you see, it's just like sulfate. I consider methyls and sulfates to be kind of two examples of molecules that are, I guess, ions that are attached to things to change their biophysical and biochemical behavior. And methyls are one of the things that are added to lots of stuff. And methylation is one of the ways that toxins are detoxified by adding methyl. So sulfate is one thing that's quite common to add. Also glutathione. You can glutathionylate, There's various special molecules that you can attach to toxic molecules, and generally to make them water soluble is what's been told. But it is also, to me, interesting that both that sulfate and that methyl that are attached to anything is valuable to the body. The body wants it. And this is the part that people are missing. The sulfate is being delivered. People never think of the sulfate as being delivered. They just think of it as detoxifying something, or inactivating. Like when vitamin D is sulfated, it's inactive. And so people kind of dismissed vitamin D sulfate because they said, well, it's inactive. It doesn't.
Is that how I store it over the winter, though? Is it a way that I can store vitamin D? Because I live somewhere where we don't have any for some of the year, or like on a shelf.
That's what I suspect. Yes, I think so. And in fact, another one is tryptophan. Tryptophan is super, super interesting. And I've been playing around with the aryl hydrocarbon receptor. Do you know about the aryl hydrocarbon receptor?
Nope.
Yeah. So for example, in the liver, The aryl hydrocarbon receptor was first identified as something that would dioxin, I think it is
Oh,
dioxin.
yes, that, yes.
Right, a super toxic thing that activates that receptor and does bad stuff, basically causing oxidative damage and all that kind of stuff. So it turns out tryptophan gets oxidized, it reacts with sunlight exposure or with hydrogen peroxide gas, either those or those in concert, turns tryptophan into something that I will call I think it's FITZ. It stands for something that's complicated, so I'm not going to be able to tell you what that is, but I think it's FITZ. And that molecule is a modified tryptophan. It doesn't even need an enzyme. The hydrogen peroxide and sunlight will do that, turning tryptophan into FITZ. And FITZ is actually the endogenous, it's the most aggressive ligand for that receptor, that aryl hydrocarbon receptor. So people now think that is the endogenous ligand for it, the thing that actually activates it for real as a natural thing. And FITZ, when it once it activates it, it causes it to turn on. It causes the cell to express an enzyme that can convert the FITZ into something else that doesn't activate it. So it's basically the FITZ turns on the production of an enzyme, and the enzyme converts the FITZ into something else, and then that something else actually gets sulfated. So it's two steps. It gets hydrolyzed and it gets sulfated, and now it doesn't affect the receptor at all. And so I actually think what's going on there, and people never look at it this way, I think that both vitamin D and tryptophan are they're basically sensors. They're trying to see if the enzymes are okay. And so if you think about FITZ is testing there's these cytochrome P450 enzymes. There's a huge class of them in the liver. Some of them oxidize cholesterol, for example, hydrolyze, I guess I should say, cholesterol, vitamin D. So they make bile acids. There's a lot of things they do, but these cytochrome P450 enzymes in the liver are super, super important. They're important everywhere in the body, but the liver has a bunch of them, and it uses them to detoxify things like these toxic, these toxic fat-soluble compounds that come in from exposures to various chemicals. They get hydrolyzed, which helps to make them water soluble, and then they get sulfated, which makes them even more water soluble, and then they can be thrown out into blood and eliminated through the kidneys. There's a class of cytochrome P450 enzymes that specialize in the sterols, which is the cholesterol, the sex hormones, the cortisol, vitamin D. Those are all sterols, and there's a class of types that hydrolyze those, and then there's another class that hydrolyze the aromatics, and that's the tryptophan, tyrosine, phenylalanine, and the neurotransmitters. All of those guys, they get modified by this other class of CYP enzymes. And so I think vitamin D is testing two things. Are these enzymes working that hydrolyze the sterols? And are these enzymes working that add sulfate to the sterols? Vitamin D tests both of those. And then tryptophan tests, are these enzymes working that hydrolyze the aromatics? And are these enzymes working that add sulfate to the aromatics? So those things are so, so important to the body that we have these specialized sensors, which are these hormones, you could say. Vitamin D is like a hormone.
Would you say vitamin D supplement is, would that be able to do the same checking, or does it have to be the biological vitamin D we make ourselves?
Yeah, that's a good question. And I have a very controversial position on vitamin D. I've never taken a vitamin D supplement my whole life. I hesitate to say that you shouldn't, because I know a lot of people have said that it's helped them. My personal feeling is that vitamin D is, so when you think about you're out in the sun and you get sun exposure and you make the vitamin D in response to the sun, but you also make vitamin D sulfate and you make cholesterol sulfate. The sun triggers the synthesis of both of those. And there's much more cholesterol sulfate than vitamin D sulfate. And the skin ships out the cholesterol sulfate into the circulation, as well as the vitamin D sulfate. So when the vitamin D sulfate arrives at the liver, it says, ding, 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 I'm here, let's check out your enzymes. Meanwhile, the cholesterol sulfate is all over the blood, it's come along for the ride. When you take vitamin D as a supplement, you don't take the cholesterol sulfate, you only take the vitamin D. So that's a false signal. That's how I see it, a false signal. So it can confuse the liver, in a sense, into thinking, in a sense. I humanize these organs, but that everything's okay, when in fact it isn't, because you don't have that cholesterol sulfate that it thinks you have because you got your vitamin D from sun exposure. So when you get it in an unnatural way, the signal's false. It's a false signal.
Because also all of these are aromatic, and because I'm really into quantum biology, the ways that the electrons are arranged is going to be different in a molecule you make from the sun versus something made in a factory. So I think the body does know the difference between, I don't care how bioidentical it is. Did you think that no matter what, there's nothing better than what we make ourselves?
Well, as I said, the deuterium becomes an issue in a lot of these molecules that we take as supplement. If you take melatonin as a supplement, you've got that acetyl, you've got that methyl, but where did they come from? Are they in fact very low deuterium? I don't think so, you know. So the whole point of the methionine is to be carrying around these super duper pieces, the acetyl and the methyl, but they're garbage because they're not right. They have all this deuterium in them. I don't know, but this is what I'm thinking. This is totally speculation on my part. So I worry about taking. So tryptophan, I would actually say at gunpoint, if I have to take something, I'd take tryptophan, because tryptophan, it's a precursor to serotonin and melatonin. But when it becomes melatonin, those things have been stuck on and they've been sourced biologically. So by the time the tryptophan makes the melatonin, it's a good guy. You know, it's got those healthy versions of those additions.
Oh, definitely. And I think it was back to what I asked right at the beginning, that if we're all exposed to glyphosate, maybe people should take tryptophan just prophylactically, just in case, because I think sort of maybe to sort of finish the story about glyphosate, because the one thing you haven't mentioned, people don't really understand, is like dioxin and DDT, doesn't it persist? Because it just comes out of the body, doesn't it? And it just goes back in and we re-eat it or re-drink it. So
worse than that, when we get it in our body, if I'm right, it goes into proteins.
Oh yeah, the glycine substitution.
And messes them up, yes. And then the protein gets broken down and the glyphosate's freed up again. It can go into a different protein. So it can wander around in your body for a very long time. I think possibly for decades, actually. But also, I think it accumulates.
Yeah, I do. Also, for people who've got things like fibromyalgia, do you think it's their collagen's disrupted because of the glyphosate?
Absolutely, you know, and we have Ehlers-Danlos syndrome is supposed to be a genetic disease, and many of the people who have it have a glycine mutation. It's been associated with many specific glycine mutations in collagen. Collagen has tons of glycine. It has a whole long sequence of GXY, where every third amino acid is a glycine. And so lots of opportunity for glyphosate's mischief. So just to make this clear, what I think is happening, and my book centers on this, as you know, I think that glyphosate it's a glycine molecule, glyphosate. It's a simple molecule, actually. It's a glycine. Glycine is the simplest amino acid. It has no side chains. Glyphosate has no side chains either. It's exactly glycine. And the only thing it has different is it has extra material stuck onto its nitrogen atom. And in fact, that extra material is a methylphosphonate unit. That's a big bulky thing. So it won't fit everywhere the glycine goes. But in the proteins that have small amino acids next door, the glyphosate can fit and put its extra stuff out there, messing up the way that protein works. And so this is how I figured out which proteins, and I verified many of them with the literature. So for example, it's been shown that glyphosate suppresses cytochrome P450 enzymes. And cytochrome P450 enzymes bind phosphate at two places. And at both places, they have 3 glycines that are highly conserved. So there's a bunch of glycines in these enzymes, these cytochrome P450 enzymes, that are critical. If you substitute something else for one of those glycines, they don't work anymore. And they're all involved in this whole idea of transporting hydrogen, but not deuterium. So these enzymes are really good at choosing hydrogen over deuterium and using sort of proton tunneling. They have some really sophisticated techniques that they use to make sure that they don't choose a deuterium.
Well, in case people don't know what proton tunneling is, because there's electron tunneling when the electrons go down the electron transport chain, but then proton tunneling, just in case people don't know what it is, because that's really important.
Yeah, it's the same thing, only electrons tunnel much more easily than protons do, because they're much bigger than electrons, but protons can tunnel. And there's a whole research space that I've been struggling to understand, because it's quite difficult biophysics, about proton tunneling. And it's really, really cool. In fact, you have these enzymes that allow water molecules to sort of pass, like in the membrane of a cell, you can have a protein in there that allows water to come through. So the cell mostly is watertight, right? It doesn't really want water to just be freely pouring in and out. But there are special places where there's kind of pores where water can come through. And they can design it so that a proton can hop through, being handed over from one molecule to the next. the proton can be handed over and come from the outside and get into the cell via this method. But but through that chain reaction, it's kind of like those isomerases that I talked about before. Deuterons can't make it through because they're too big, they're too bulky. You know, they don't easily come through. It's a very narrow channel. It's a water string, really. It's really cool.
Oh yeah, proton wire, yes.
Proton wire, yes, it's really, really cool. And I think those water strings are absolutely central for, did Jerry Pollack talk about those at all?
Yes, of course he did. Yes.
Yeah, because those are so cool. And you basically, you've got, the water has a hydrogen, an oxygen with two hydrogens attached to it. And then you can have H3O plus with an extra proton. And you can have OH minus with a missing proton. So what happens is you have H2O over here on one end and you stick in another proton. Now it's H3O plus. So it wants to get rid of one of its protons. So it hands it over to the next guy. And then he's H3O plus and he hands it over. So the proton trickles through the water chain and makes it to the other side. So these water chains are set up inside certain enzymes that are very carefully constructed to allow the water to get through. They're really fascinating. And there's that sort of thing going on in the cytochrome P450 enzymes. They have like a dozen water molecules that can get into this hydrophobic space. And the dozen water molecules can line up and then help the proton to go from one ring. It's coming from a ring usually. And that's why you have these rings in those two classes of molecules that I mentioned that are so important. The serotonin class, which is the aromatics, and then the sterols, which is the cholesterol and the vitamin D. So those two classes are really important. They all have rings. They usually have a six carbon ring and a 5 carbon ring. And I think that that's super important for, I suspect, for influencing gelled water. That's a whole other topic we can get into, but that's the Gerald Pollack specialty, the gelled water, which has these six water molecules. So the 6 carbons are kind of like six water molecules.
Like a hexagon, yeah.
It makes something similar, and I think it kind of triggers the gelling of the water. So I think these aromatic, these two classes of molecules are super, super important in biology. I think a lot of their importance has to do with two things. They can transport sulfate and they can help to gel water. They can catalyze gelled water and energize the mobility of the protons in the gelled water. Oh, I'm suspecting this.
Yeah, because I suppose people know it as EZ water or exclusion zone or structured or live water. Because one thing I've wanted to ask you is, we're going to talk about aging, but with sulfate, how do we lose it as we get older? Where does it go? And because it's obviously a really important part of aging that people like Aubrey de Grey don't mention, same as deuterium, but where does it go?
Yeah, well, that's the whole problem. We can't transport it because we can't sulfate these molecules. That's a huge, huge issue. When you've got problems, and those enzymes that sulfate, I think they're being disrupted by glyphosate. They have these glycine dependencies, and they also, I talk in my book about various reasons why I suspect that the sulfotransferases are being disrupted. Also, the one that makes this energized sulfate. There's a step that is that produces it's a molecule that has sulfate sort of plus an ATP molecule. It starts with ATP and sulfate, puts them together, loses one of the phosphates, and makes this PAPS, PAPS, phosphoadenosine phosphosulfate. Kind of a complicated word. Yeah. PAPS is a sort of energized sulfate molecule, and that's the universal sulfate donor. So anytime you take a sulfate off of some molecule, you make PAPS. You have to make PAPS to make it, to be able to move it to something else.
The SAMe of sulfate, so it's kind of neglected. Oh, I get it. So the PAPS. So for people that want to get more sulfate into themselves, what can they do? Because I mean, that San Pellegrino's got lots of sulfate, and
yes, I think that's good actually. There was a fascinating study that was done by it was published with looking at water and autism. It was quite interesting. This person actually contacted mothers of autistic kids and figured out what kind of water the kids were drinking and looked at the water in the different states and how much sulfur, and got all the different bottled waters, how much sulfur. He had it all figured out. And he showed there was a statistical correlation between drinking water that's low in sulfur and having autism. It was quite amazing. I couldn't believe that one factor could actually play a statistically significant role.
But
It's pretty amazing.
that's massive. Well, what about MSM and sulfur-containing
Methylsulfonylmethane, yeah, that's good. And also there's DMSO, dimethyl sulfoxide.
Oh, I love that.
There's other sulfur-containing molecules too. I like garlic. I eat tons of garlic. Garlic and onions are both good sources of sulfur. I always try to go for fruits and cruciferous vegetables. They have a really good.
Leeks as well have got.
Leeks are wonderful. Yes, all of those
Brussels
onion
sprouts,
family.
yeah.
Brussels sprouts, yes, those are great. We eat tons of onions, garlics, leeks, and then cauliflower, broccoli, Brussels sprouts, all of those. That's a staple in our diet in my family. So I really love those. I like them to begin with. They're tasty, but they're also, I think, really important for the sulfur. So you can eat foods that contain sulfur. I also recommend eating foods that contain tryptophan. So that's because of the tryptophan problem with glyphosate. So I like to eat nuts, and nuts are a good source of tryptophan, also seeds and nuts, and also chicken and turkey. So there's some animal-based sources as well. And of course, sulfur is also good in animal-based sulfur. So eggs are really good, organic eggs. And meats, they have a high quality. Cysteine is a really good source of sulfur. It's a sulfur containing amino acid. Of course, you have N-acetylcysteine, S-adenosylmethionine. Those are both sulfur containing. And also lipoic acid. That's another thing. People take a lot of these. I also find chondroitin sulfate, glucosamine sulfate. People think they're taking glucosamine, but I think they might actually be taking sulfate, if you know what I mean.
Oh yeah, I know. I think some people think that glucosamine binds to lectins, and that's why it prevents arthritis. But it could just be the sulfate, same as the Epsom salts. Maybe it's the magnesium,
exactly a magnesium sulfate. People all think, oh, for the magnesium, but I always think of the sulfate. I think there's many cases where people don't realize the importance of the sulfate, because sulfate, it's not just a matter of getting sulfur. It's a matter of having those enzymes working properly, and that's where the glyphosate comes in. So if you're being chronically exposed to glyphosate, people get in trouble. And when I first started talking about the importance of sulfur, I got email from people who said, I can't eat sulfur-containing foods. I get sick. I have sulfur sensitivity. A lot of people complained about sulfur sensitivity. That's, I think, because of the glyphosate messing up those enzymes, because then you get sulfite. You can't go to sulfate. So sulfite builds up. You have these sort of sulfites in wines and stuff people can be sensitive to. The sulfite gets reduced by gut microbes to hydrogen sulfide gas. And then that gas can become very disturbing. You get bloated, and it can also travel up to the brain and give you brain fog. A lot of the autistic kids, I think, are suffering from hydrogen sulfide gas. That gas probably has low deuterium, just like hydrogen. That's what I'm thinking. Hydrogen sulfide produced by the gut microbes, the same thing, because deuterium likes to stay in the liquid phase. So when you make a gas, you're more likely to have hydrogen in the gas.
So if you wanted to make deuterium depleted water at home, would you freeze it?
Yes, that's an interesting thought. And I should mention that because the water that comes off of glaciers, I don't know if you know that, is low in deuterium.
Oh yeah, in Siberia, yeah, where they discovered the deuterium because of the centenarians.
That's right. That was so fun. I really loved that story. That was how that was first figured out. The Russians were really leaders in that space, and it's still the case that it's mostly Eastern Europe and Russia where the research is going on with respect to deuterium. Americans are pretty clueless. I mean, the whole American medical system has no clue about deuterium. You'll never see anything about it as far as I can tell. Maybe some of the alternative specialists are starting to pick up on it, but it needs to be out there. I think people need to be aware. And it's it's not just eating low deuterium, it's making sure that those enzymes that manage deuterium are working properly. That's a critical thing. And we didn't talk about NAD, but we should have nicotinamide adenine dinucleotide. Sometimes people can take that as a supplement, I believe.
Oh, like NMN and NR, like a precursor. People have IV NAD plus treatment as well.
Exactly. So NAD, that comes from tryptophan, and tryptophan, of course, is blocked by glyphosate. So that's another problem. The tryptophan is a precursor to NAD. And of course, niacin is also along the way there. So eating foods that are rich in niacin is important. And also the flavoproteins, the flavins, riboflavin, because the flavoproteins, the ones that glyphosate disrupts, they depend on flavins, which come from riboflavin. And that's another B vitamin. So the B vitamins are really hit hard by glyphosate. And cobalamin is messed up too in different ways. I've written about that by glyphosate. Cobalamin is very tricky to get in because it's a big bulky molecule, and it has to be special escorts, and those things get messed up by glyphosate. So all the B vitamins are in trouble with glyphosate, which means that for one thing, you've got to eat foods that are high in B vitamins.
But that's really interesting because you can put in as many precursors as you want, but if the molecular machines, the enzymes are not working, there's no good. Because what could people say if somebody's got lots of glyphosate in them? How do they get it out?
Yeah, there's a good question, and some people have offered solutions that I think could be working. It's not clear, but I know that there was this product called Restore. Zach Bush was promoting this Restore. It contains fulvic acid and humic acid, and also minerals, and then probiotics. I think that kind of mixture, minerals, probiotics, fulvic acid, humic acid, can be useful. Of course, the minerals, we didn't get into that. We intended to, but we'll have to
that's
do
my
another
favorite subject, because
day.
I was gonna say, would Sheila G help as well? Because that's got sort of trace minerals in it as
Yes,
well. And
I've
then
heard that's good. And
what
also bentonite clay and carbon, and also fermented foods. I have been claiming that fermented foods could provide microbes that can completely break down glyphosate. There are a few. Glyphosate's a tough molecule to break down because it has this CP bond that's really difficult. And so there are a few microbes that can break it down. And there are species of Acetobacter that can break it down. So if you have those, if you're lucky to have those particular strains of Acetobacter in your probiotic, it can break down the glyphosate before it even gets to your stomach, I suspect.
would that be
So
in live apple cider vinegar?
apple cider vinegar, sauerkraut, yeah, even fermented dairy like cheese and yogurt and those kinds of things.
Oh, and kefir. Okay, I see. Yeah,
Yeah, right.
because before we wrap up, because is there anything else you can say about minerals? Because things like anything to do with fluoride or iodine or manganese, because I know you had a paper on
Oh yeah, I mean, I could say a lot
manganese
about all
gets
of them. I have a
neglected.
whole paper on manganese that I wrote
I know,
together
because
with
I really love
Anthony St.
manganese and people don't talk about it.
Yeah, what I will say is that glyphosate makes minerals both toxic and deficient at the same time. And I know that's true for manganese and iron, and probably for other things as well. And the paper was all about manganese. It's quite fascinating. Glyphosate is a really good chelator, so it grabs hold of the metals and doesn't let go. And the lactobacillus critically depend upon manganese to stay healthy. And glyphosate deprives them of their manganese, and that's part of the way in which glyphosate kills them. So the lactobacillus are very sensitive to glyphosate. That's been shown experimentally. And they're crucial in the baby's gut for drinking milk. So I think that's another problem with a lot of kids having issues with milk. A casein intolerance, they have to go on soy, and soy is loaded up with glyphosate. So it's just a downhill path from there. So
isn't there another twist to the manganese and the glyphosate? Because doesn't it end up accumulating in your brain because of the glyphosate, and it could give pseudo Parkinson symptoms?
exactly, yes. And there's a condition called manganism that's associated with welders, because they're exposed to the manganese when they weld, and then they get this. It essentially looks a whole lot like Parkinson's disease. Manganese is an unusual mineral in that it travels very well along nerve fibers, and so glyphosate delivers it to the liver, and then messes up the liver's ability. Normally it gets attached to bile acids and sent back to the gut, and eventually distributed to the body, actually, because the body wants manganese. It's a healthy mineral. But that gets blocked by glyphosate because it messes up those cytochrome P450 enzymes that are needed to make bile acids. So the bile acids become deficient. The manganese sticks and stays there, goes into the gallbladder and the liver, and then eventually gets shipped up to the brain along the vagus nerve. So I think that's what's going on. And so it accumulates in the brainstem nuclei, and then you get nasty conditions like Parkinson's. Probably even contributes to autism, but I'm not sure of that.
Oh, that's really important, because people are very interested in the vagus nerve now. And that's why, because people might think, oh, if the lactobacillus needs manganese, I must take more manganese, but then you'll end up dumping it all in your brain. So there's no matter what, you have to know the mechanisms of these things, and it's very intricate. So that was really interesting.
Yes, that's so true. And I think the same for iron. We've got a mess right now with iron. We've got iron deficiency and iron toxicity simultaneous, even in the same person, both, because the iron is another mineral that the body has really sophisticated mechanisms in place to distribute these minerals. They're very important where they're needed to catalyze various enzymes, but the body knows how to carefully usher them to the place where they need to go. But those mechanisms, just like with the sulfate, they get messed up by glyphosate, and then they become toxic at the same time as they're deficient. So you're taking more because you think you're deficient, and then you can actually ruin your liver with iron toxicity.
Oh yeah,
It's a
that's why
nasty situation.
Yeah, iron labs are a nightmare to read. Where do you think actually the real root cause is? Just look at glyphosate instead, and if the glyphosate's high, then you can
I think
discount
so.
all of those iron labs. Because I've pulled my hair out over them, because sometimes they don't make any sense.
Exactly. I think that's what people are not realizing, the importance of having the mechanisms in place and working properly that distribute these things. Anything, and oxygen too, of course, oxygen is essential to life, but also very toxic. And glyphosate causes you to be unable to manage oxygen correctly. And that's why you get all this oxidative damage. So it's a nightmare. I mean, I think I wish we could just ban glyphosate worldwide. If I were king, that's what I would do, or queen, I guess. I would ban glyphosate everywhere on the whole earth.
Do you think it's worse then than dioxin and DDT?
I do, but not necessarily because it's more toxic, but because it's more prevalent. I think it's just everywhere, and people think it's safe. So people are using it carelessly in their yards, and it's all over the food supply. The government doesn't care. There's a lack of awareness of its power that's making it much more pervasive and much higher exposure for a much greater population.
Yes, definitely. We need reminding. But anyway, so thank you so much for sharing your giant brain. It's been fascinating. Like always,
that was quite a chore. I think people will have a hard time.
Every time I listen to you, I always learn something new. So it's been really, really interesting.
We'll have to do this again because there's lots more to say. This was
oh
fun.
no, I know that's the thing. And definitely, I highly recommend the book because each chapter is like its own little book. So you don't have to absorb it all in one go, because there's the phosphate puzzle is one of the chapters, and that's really interesting as well. But definitely the book's a really, really good read. So thank you very much.
Okay, great to talk to you. Yes, we'll have to do this again.
Yes, definitely.
I love talking to someone who actually knows a lot, which is the case with you. So